Hey everyone, let's talk about nucleic acids. They are for information storage, and you probably recognize the molecule DNA because DNA is a nucleic acid. So this is our last of our macromolecules. So we have a test coming up very soon, and I let you know when that test is in class. So which one are you? Which picture represents you? Nucleic acids, they're for information storage, and DNA and RNA are our most popular of the nucleic acids. The function is that they are genetic material. They store the recipes. DNA stores the recipes to make the proteins that are in our body. So we can say that our genes are like the blueprint for building proteins. If we remember how it works, that the DNA is stored in the nucleus of the cell. RNA is the other nucleic acid molecule that comes in and makes a copy, or what we call a transcript, of the gene, and then sends that message out through the cell nucleus to the ER, through the little tubes that make up the ER, and then finds a ribosome, which could be on the ER or floating out in the cytoplasm. And that ribosome locks onto that RNA transcript, which was a transcription of the gene from DNA, and then puts together the correct amino acids to make a polypeptide, which then will eventually go on to be shaped correctly to become a protein. So that's how that works. So that information then can build the proper proteins for the cell or sent to other cells and can be transferred to the next generation from you to your kids. So DNA and RNA. RNA is ribonucleic acid. It's the single helix molecule, and DNA is the double helix molecule. Both of them are considered polymers as they are very long molecules made out of repeating units called monomers, and the monomer is a nucleotide for both of them. A nucleotide has three parts. First of all, there's what's called the nitrogen base. That's this part right here. It has, as you guessed, a lot of nitrogen in it. Lots of carbon, lots of nitrogen. Connected onto that is a sugar, a 5-carbon sugar. It's called, if it's RNA, ribose, and if it's DNA, deoxyribose. Got that OSE ending because it's a sugar. So that's here, and then off the end is a phosphate group. Remember the PO4 group. Also, it gives DNA or RNA a negative charge because of that phosphate group. So remember, there are 4 different nucleotide monomers that you can find in DNA. They are A, T, C, and G. And then the 4 nucleotide monomers that are in RNA are A, C, G, and U. You remember what those stand for. We'll get to that. And here they are. Get me out of the way. The adenine, the A, and the G, guanine, those look like this. They are basically a double ring structure, and you can see the shape of them. They are classified as what we call purines. For the other 3, cytosine, thymine, and uracil, you'll notice they all look very similar because they have a single ring that makes them. They're all classified as pyrimidines. A good way to remember this, at least what I say in my head to help me remember, is pure silver. So I know that pure, and then silver's symbol is Ag on the periodic table, so pure silver, purine. That helps me remember that A and G, adenine and guanine, are purines. Just a little trick that might help. So when we make a nucleic polymer, we have a backbone. And so we can see how the sugar is connected to the phosphate group here. And then a bond between them, which is called a phosphodiester bond. So I'm going to play this so that you can see that this is our phosphodiester bond there. And remember, each nucleotide is circled, containing the nitrogen base, the sugar, and the phosphate group. And then in red, circled is the phosphodiester bond. So the phosphate and the sugar are considered the backbone to DNA or RNA. So each new base is added to the sugar of the previous base, and it grows in only one direction, which we'll get into further when we get to those chapters. Notice when they come together, DNA is double-sided. When they come together, A's and T's join together because they like to have 2 bonds between them. G's and C's join together because they like 3 bonds. And those are hydrogen bonds in between the 2 sides of DNA, which is good. They're weak bonds, but we want them to be weak so that we can separate them whenever we do cell division, like mitosis or meiosis, or when we need to transcribe a gene into a protein. So they always join. If there's an A on one side, you'll always have a T on the other. A G on one side, there'll always be a C on the other side. So it makes it very predictable. Oops. And so there's what DNA looks like. It's twisted as a double helix with the nucleotide monomers across from one another, G's with C's, A's with T's, and hydrogens in between. When we copy DNA, that's called replication. We say the strands are complementary to one another when that happens. So we separate our DNA, or sometimes we say we unzip it, and then we fill in what's called complementary bases. So if I unzip this and I have a G on one side, then I need a C on the other. If I have an A on one side, I need a T on the other. So basically, if I have one side of DNA, I can rebuild the other, which is basically what happens during replication as both sides separate from each other, and then you have to rebuild a daughter strand for each old parent strand. So this tends to be a good system. DNA is a very small molecule. We're talking nanometers, and nanometers, remember, is 10 to the negative 9. Very small. It's got to fit into the nucleus of our cells. DNA gets copied during mitosis and meiosis. Mitosis is just normal cell division, and meiosis is cell division that creates sperm and egg, or what we call gametes. This was discovered by Watson and Crick. I could put myself in the middle there. Oh, no, maybe not. Watson and Crick, and they used research from other researchers basically to figure out that DNA was a double helix. There were other tests that already confirmed that there was guanine, cytosine, adenine, and thymine in there, and then the sugar and the phosphate groups. That was already known. But all that other research then helped them figure out that this is in a double helix. And then they went and built this little model here. Oops, I'm sorry. Looks like a cool little model. And there's Watson and Crick there. And they won a prize, the Nobel Science Prize for this. This is another researcher from England that was involved, and another researcher from England, although she died, and they don't award these awards to anyone who has passed on, so she didn't get any credit for that prize. But her X-ray diffraction picture of DNA, a little mathematical equation with that X-ray can show that DNA is a double helix, and they used her data as well. So we'll give some kudos to Rosalind Franklin there. Some interesting information. That ratio of A's to C, or A's to T's and G's to C's, helps to show the stability of the molecule. Organisms that live at high temperatures have more G's and C's. And parasites tend to have more A's and T's. And we're not quite sure why. It might be something to do with having 3 hydrogen bonds between the G's and C's and only 2 between the A's and the T's. That's an area of research there. And here's another little interesting note. While DNA and RNA are big, long polymers, ATP is also a nucleic acid, but it exists as just a single molecule at a time. It's a little bit different in that it still has adenine, it has ribose, so that's similar, but instead of 1 phosphate group, it has 3. All right, that's why we call it ATP, adenosine triphosphate. And remember, whenever we have bonds, we have potential energy. So your body, your cells, will break the bond off this last unstable phosphate group, and will use the energy released from that to move things and make things happen in your cells and in your body. So there's also GTP, guanine triphosphate, and other ones, but ATP is the one used most. And again, it is also considered a nucleic acid. I want you to make sure you get to know the ATP molecule. You should start practicing drawing it. Maybe not so much like this one up here, but one like this with the nitrogenous base as maybe a hexagon. That'll work. And then a pentagon for the sugar, and then 3 phosphate groups on the end. That way you know all about ATP and how it looks, and in a further chapter down the line, when we really work more with ATP, it'll seem more familiar to you. And there's the double helix. As I said, we weren't going to go too deeply into nucleic acids just yet, because we'll do more with them further down the line in a chapter to come.